Okay, welcome to our weekly live online session. So it's week 13 of our course, and I believe that we're really looking at the content of the chapter on central nervous system. So let's make a start with looking through some of the key points of this chapter's content together. Um, I do want to real quickly to create that space for checking in. I also want to kind of outline what's the plan for the week, especially when it comes to the homework that I'm looking for. And then we'll review the highlights of the weekly chapter material. This week sometimes confuses students a little bit since the content will be mainly from chapter 14, but there are certain elements of the content that also relate to the chapter 13. So you might need to kind of browse back and forth a little bit with those chapters. So just something to kind of get you thinking of that as well. And as I've done before, I've started these weekly sessions with a quote from the book by Pueblo Inward. And the quote that randomly came up was for this week was that peaceful minds have the power to create a peaceful world. And I really think that what this shows us is the important role of the central nervous system. We end up seeing that central nervous system is really what creates us and the experiences of the world that we have. So I think that that's a nice bridge to our content. For this week's homework, I'm asking you to complete two assignments. I'm asking you to look at the chapter 14 quiz and also the chapter 14 practice anatomy. Both of these are due at the end of the week on Sunday. I believe it's going to be 17th. But as always, please do check your syllabus and your Canvas course. It outlines those there as well. But kind of a manageable workload, I believe, for this week. So hopefully it will give you a chance to enjoy actually of learning the material and digging into it. So what I wanted to start our review with is a quote that how one of my students once described this chapter to me, and their description was that the brain is nothing but a lump of fatty tissue. I think that that is an interesting thought. Maybe it a little bit also underestimates the fact that brain is and a creation of the brain is really where we get all of the characteristics that make you uniquely you. Your personality, your memories, your dreams, hopes, imagination, wishes, and future thoughts all are a creation of your central nervous system and especially the brain. So I do think that that's an interesting thought and something to keep on mind. So I think one of the questions that we can kind of ponder on as we think of individuals being unique in terms of the creation of their brain is also how did we end up here? And nearly all animals have some kind of a nervous system, whether it is from earthworms, very simple nervous system, to highly sophisticated nervous system of, for example, dolphins. What we see is, though, that most animals, once we move to a little more complex animals, do share some similar parts and similar regions within their central nervous system. And we will be focusing on our course, obviously, on the human brain, but we'll also do occasionally comparison to the sheep brain, especially when my students in face-to-face classes would be doing dissections. And I think I have some examples also on your online course material of that. We see that we have similar structures. The proportions and the shapes of these structures might vary. Remember, sheep have very different physiological needs than humans do have. But we do use comparative anatomy for the learning process and often do those dissection activities using animals because that's something that's affordable and accomplishable for us in this setting. I think that another interesting theme or interesting concept to talk about is how did we end up here also in terms of evolution? And over the past 3 million years, there's been significant changes. What we saw is that the human skull grew in size, but also the size of the human brain grew. And what we're really seeing is that the biggest changes took place on the anterior portion, so on the frontal lobe of the brain that grew. And we also saw that the human brain also evolved in a sense that we had more cortical area, more cortex. And this was being able to facilitate as the size of the skull could grow only so much by making sure that we formed more folds. So that way we were able to fit in more surface area. And a good analog that I often offer for my students about that is that if I have this large surface area of a piece of paper, and now I want to fit this large surface area in as small of a space as possible, what I can do is crumble that paper and I still have the same surface area, just packed more compactly. And I think that that's one of the things that we saw with the development of the cortical regions of our brain. Other question that's sometimes kind of interesting to ponder with is what happens to us during a lifetime of an individual. And what we end up seeing is that brain is one of the, and nervous system is one of the first systems to start to developing, starting from week 2. And it does grow at the fast rate during the prenatal and early life, and then slows down the growth. Do notice though that even though the size-wise we might reach a peak with the size of the brain, there is always constant changes taking place. For the longest of time, we did teach to our students that your brain is not able to generate new neurons, and that is largely true. So that's why activities that damage your neurons, like consuming large amounts of alcohol, can be problematic. We're seeing some modest neuron regrowth taking place as well, but it definitely isn't enough to compensate for the loss of neurons. So we want to keep that on mind as well. If we think of the embryology, if we think of the development of the human nervous system and especially the brain, what we see is that the brain and spinal cord start developing as a neural tube. And within that neural tube, we can see certain areas that will then later take the shape and take the role of specific parts of the brain. So let's have a look at that together. There on the left-hand side, we're seeing this early neural tube that appears and how it slowly starts to expand and shrink in certain parts, and then it starts folding. And these different parts that we see here in the early development of the brain are going to be reflected as different portions of the brain later on when we look at the adult brain. So something to keep on mind about those regions as we study them throughout our course and throughout this chapter. So if we look at the adult brain here, really there's a few areas for us to pay attention, and we'll see the process that leads us to those, as most of you might know them in an adult brain. So this is a 5-week after fertilization, so early development. You see that very much of a neural tube, how it starts folding, how certain parts start extending. And we see the same brain at 13 weeks post-fertilization in the development. And at this point, we're already starting to see a lot of resemblance to what the adult brain will be in its shape. And here we see the brain at the birth. So we can see very familiar structures for us that you might be already familiar with. We see our cerebral hemispheres. These are the big parts, what we see on the outside. So there's kind of a faded portion that's showing us what would be inside these cerebral hemispheres. We also see our diencephalon buried inside the cerebral hemispheres in the middle. We're going to see our brain stem shown green on this diagram. This is actually the first part of the brain to develop, and this houses many vital functions for sustaining the life, really important things for our survival. And finally, we see our cerebellum, which is also sometimes described as a little brain at the very back. So we will go through these different parts of the brain. Before we do so, though, I think it's a good place for us to start by looking at the different portions of the brain when it comes to the neurons and nervous tissue that we've just discussed about. And you remember that some of the nervous tissue could be myelinated. So there was that fatty tissue around those axons, and some is non-myelinated. And what's interesting is that we see often the cortex, the outermost layer of the brain, being gray matter. So this is the matter that has a lot of cell bodies, non-myelinated structures of the brain. So really the processing part of the neurons. And the white matter, which is more centrally, more medial parts of the brain, medial parts of the cerebral hemispheres, tend to be myelinated axons. We see other tissue there as well, but it's largely information pathways for passing signals from one portion to another. If you're a fan of Sherlock Holmes books, you might remember that Sherlock Holmes always says that he needs to dig into his gray matter. That means that he's doing a lot of thinking as he's trying to solve these murder mysteries. So that would be the cortical region of the brain. What I'm showing there on the right-hand side is a cross-section through spinal cord. And rather interestingly, here we see this flipped other way around, that the gray matter represents the inner portions of the spinal cord, whereas the outer portions are going to be white matter. So something to keep on mind in relation to that. Other thing that I really wanted to kind of show at this point is the empty spaces within the brain. And these empty spaces are filled with fluid known as CSF. This is our cerebrospinal fluid, and it bathes the brain both from these empty spaces inside, but around the brain as well within the skull. These empty spaces are what we call ventricles. And remember from our earlier discussion, I believe last week, when we had a look of glial cells, these are lined with ependymal cells. So that's interesting and noteworthy to keep on mind in sense. Let's move onwards. Let's have a look of our cerebral hemispheres a little bit more. This represents about 83% of the total brain mass. Some terminology for us to see here. We're seeing the term uri refers to these ridges or bumps on the surface of the brain. We're seeing sulci, which are these shallow grooves of the brain, depressions. And then we have fissures, which are very deep, very prominent grooves. And often we use these fissures to label specific parts of the brain. So here we can see our longitudinal fissures separating the left and right hemisphere of the left and right cerebral hemispheres. So we're seeing here nicely the longitudinal fissure. We can also see on this picture our transverse cerebral fissure separating the cerebellum and cerebrum from each other. So again, that terminology that you learned early on the course definitely comes in useful at these later stages of our study material. We can see a couple of other important sulci that we use. We see our central sulcus running kind of in the middle of across kind of on a coronal plane. We can see our lateral sulcus that separates then a lobe there. We can see our parieto-occipital sulcus, and finally our transverse cerebral fissure. And the reason why these depressions are important for us to know is that they help us then to understand the different lobes that are formed within our cerebral hemispheres. So let's have a look at those. First, we're going to see our frontal lobe. We're seeing the frontal lobe often holds things like processing, things like thinking, planning, executive functions. And right at the posterior portion of the frontal lobe, we're also going to see our primary motor area located there, as I'm highlighting it. Our parietal lobe then contains a lot of sensory processing, and again we see primary somatosensory area right next there at the anterior portion of the parietal lobe. Occipital lobe typically is dedicated for processing vision, and then temporal lobe often deals with things like language, hearing, and speech. So lots there for us to know. The insula that I have in brackets is actually embedded here deeper between our temporal and frontal lobe. So you would have to dig in there to see there. So one of the questions that my students sometimes have is that how do we know these different functions that are associated with different areas of the cerebral hemispheres? And really the answer to that is thanks to brain injuries. We talked about the man who mistook his wife for a hat, and we talked about Phineas Gage last week. So that's been great source of understanding what's located at what part of the brain, but also from brain imaging studies. So we're able to do a lot of that, and that used to be my job. So we're seeing that certain areas of the brain become active as the patient is performing a particular function within the functional MRI imaging scan, for example. And a couple of different specific areas that I wanted to highlight from this diagram, in addition to what we've seen. We're going to see our Broca's area, which is a lot to do with speech production, and our Wernicke's area, which is really speech comprehension that takes place. From this picture, again, I'm showing the primary motor area here at the back of the frontal lobe and primary somatosensory area here at the front of the parietal lobe. Notice how these lobes were named after the bones of the skull. So again, putting in that effort to learn those is definitely, hopefully, paying off at this point. We sometimes also talk about lateralization of the cortical functions, so division of the labor on both hemispheres, left and right. Some people are characterized as being left brain dominant or right brain dominant. There's some debate about this approach. But I'll let you dig into more of that if that's of an interest to you. Often we describe that right hemisphere would house things like language, math, and logic, where the left hemisphere is for things like visual, spatial, intuition, emotion, artistic, and musical abilities. But remember that this lateralization is not fully accepted, not fully proven, so there's some debate on that as well. What we see here, moving away from the cerebral hemispheres, next I wanted to talk about a structure known as diencephalon. That's the part inside that we're going to see. And we'll talk also about the brainstem, the portion that then comes out. There are many nerve tracts with white matters present as well. But let's focus on this diencephalon a little bit. And within diencephalon, we really see three important functions. We see our thalamus, we see our hypothalamus, which literally means underneath the thalamus. And then we see also our epithalamus, as pointed there on this picture. If we try to associate certain functions within these regions, we see that thalamus is kind of this egg-shaped blob on both sides. There's a lot of fibers that communicate with the cerebral cortex. cortex. So we often think of that as a relay station for information coming in and coming to the cortex, things like that. What else? It sorts, it edits, it relays information. So a lot of that taking place: emotions, visceral functions, direct motor functions, memory, sensory integration, and so on. From the hypothalamus, like I mentioned, that's the portion below the thalamus. For example, we see smell relations taking place there. And the reason why I wanted to highlight hypothalamus for us is that we see that that's what controls our pituitary gland. And we'll talk about pituitary gland in great extent when we get to the BIO202, a really important, known as a master gland of your body controlling other endocrine glands. So an important structure for us to talk about and learn about. When I'm thinking of what else could I share with you, I guess that we could also see a lot of regulation of the homeostasis, for example, taking place, controlling our autonomic nervous system, blood pressure, heart rate, digestive tract, pupil size, physical responses to emotions, regulating our body temperature, regulation processes like sweating, shivering, hunger, feeling full, thirst, and so on. We really see that endocrine system function control through that pituitary. And finally, our epithalamus. From there, I really wanted to highlight the pineal gland. It produces melatonin, regulates our sleep-wake cycle. We'll get to see a little bit of that when we look at the dissection of the sheep brain as well, when we're talking about that. Let's keep going on with our review of the central nervous system. And next set of structures that I wanted to talk about include our brain stem. So shown green on this diagram here, there's really three parts: our midbrain, our pons, and our medulla oblongata. Midbrain is located inside the brain. Still, pons is this first bump that you can see, and then the medulla oblongata is like the second blob, if you wish to think about it. This is very much like spinal cord, these regions, but housing vital functions essential for our survival. Like I mentioned, midbrain serving as a relay station for a lot of information. Pons, for example, regulating breathing rhythm, and then with the medulla, we see things like controlling our equilibrium, relay station, reflexes, heart rate, blood vessel diameter, breathing, things like hiccups, vomiting, swallowing, coughing, sneezing. So a lot of things going on there. And finally, I wanted to finish off by pointing out the cerebellum, so the little brain as we know it. This represents about 11% of our brain mass, and this region is really dedicated for controlling our movement, controlling the balance. And if we do take a cross section through the cerebellum, we will see this white myelinated axons running in there. They look a little bit like branches of a tree. So that's why we often describe this structure, these white branches, as arbor vitae, which refers literally to the tree of life. So something interesting there to note as well. And I think that about brings us to the end of the key structures from this chapter in terms of reviewing those. Of course, I encourage you to do the homeworks as well. I think that the homeworks follow so closely of what you have learned in the past that I'm not going to go through them again, but I really encourage you to look those as well. And in the next chapter, we will pick up from here and continue our review of the nervous system. At this point, I think this is a great place for us to wrap up. As always, I appreciate you being part of this class and look forward to seeing the great work at the Canvas, working with you on the forums, and seeing where this week's material takes us. So I look forward to that and I hope you have a good week. Please remember, as always, I am here to help you. All you have to do is reach out to me and we'll figure out if there's anything that I can assist with, or even if you just want to share your insights, aha moments.